Tissues. All living organisms are made up of cells. In unicellular organisms, a single cell performs all basic functions. For example, in amoeba, a single cell carries out movement, intake of food, gaseous exchange, and excretion. But in multicellular organisms, there are millions of cells. Most of these cells are specialized to carry out specific functions. For example, in human beings, muscle cells contract and relax to cause movement. Nerve cells carry messages. Blood flows to transport oxygen, food, hormones, and waste materials. In plants, vascular tissue conduct food and water from one part of the plant to other parts. Cells specializing in one function and often grouped together in the body. This group of cells is called tissues. A group of cells that are similar in structure and work together to achieve a particular function forms a tissue. Are plants and animals made of same type of tissues? No. Plants and animals do not have same type of tissues. They have different types of tissues. Plants. Plants are stationary or fixed. They don't move. Since they have to upright, they have a large quantity of supportive tissue. The supportive tissue generally has dead cells. Animals. Animals, on the other hand, move around in search of food, mates, and shelter. They consume more energy as compared to plants. Most of the tissues they contain are living. Textual questions. What is a tissue? A group of cells that are similar in structure and work together to achieve a particular function forms a tissue. Second question. What is the utility of tissues in multicellular organisms? The utility of tissues in multicellular organisms is to provide defined structure, mechanical strength, and division of labor. This division of labor makes multicellular organisms able to perform all functions efficiently. Now, let us learn about the plant tissues. Flowchart of the plant tissue. Plant tissues are mainly divided into two types. First one is the meristematic tissue and second one is permanent tissue. Meristematic tissue is again divided into three types. First one is the apical meristem, second one is intercalary meristem, third one is lateral meristem. Permanent tissue is divided into two types. Simple permanent, complex permanent. Complex permanent tissue divided into two types, xylem and phloem. Simple permanent is again divided into three types, parenchyma, collenchyma and sclerenchyma. Parenchyma again divided into two types, arenchyma and chlorenchyma. Activity. Growth of roots in onion bulbs. Take 2 glass jars and fill them with water. Now take 2 onion bulbs and place one on each jar. Observe the growth of roots in both the bulbs for a few days. Measure the length of roots on third day. On day 4th, cut the root tips of the onion bulb in jar 2 by about 1 centimeter. After this, observe the growth of roots in both the jars and measure their lengths after 5 days and record the observations. Observations. Jar 1 onion bulb has longer roots than jar 2 because the jar 1 onion root tips are not removed. The growth of plants occurs only in certain specific regions. This is because the dividing tissue known as meristematic tissue. New cells produced by meristem are initially like those of meristem itself, but as they grow and mature, their characteristics slowly change and they become differentiated as components of other tissues. First type of the cells, meristematic tissue. Meristematic tissue is made up of a group of cells that divide continuously to form new cells. This tissue is found in the growing regions of plants, such as the tips of roots, stems, and branches. The function of meristematic tissue is to make plants grow. The cells of meristematic tissue are small and cubical. They also have thin cell walls. They are tightly packed with no intercellular spaces. Cells of meristematic tissue are very active. They have dense cytoplasm, thin cellulose walls, and prominent nuclei. They lack vacuoles. On the basis of position in plants, meristematic tissues are mainly divided into 3 types. First one is apical meristem. Apical meristem is located at the tips of the roots and stems. These cells are responsible for the increase in length of the plant. Second one is intercalary meristems. These tissues are located at the base of the nodes where leaves are attached. They are responsible for stem elongation or longitudinal growth. Third one is lateral meristem. The tissue is found along the lateral sides of the stems and roots. The girth of the stem or root increases due to lateral meristem. Permanent tissue. The tissues that are completely grown and have lost the ability of division are known as permanent tissues. The meristematic tissues divide and differentiate to form the permanent tissue. Differentiation leads to the development of various types of permanent tissues. Types of permanent tissues. First one is simple permanent tissue. A few layers of cells beneath the epidermis are generally simple permanent tissue. Simple permanent tissue is further classified into 3 types. They are parenchyma, collenchyma, sclerenchyma. Parenchyma. Parenchyma is the most common simple permanent tissue. The cell wall of the parenchyma is thin and made up of cellulose. They are living cells. They are usually loosely arranged, thus large spaces are present between cells, intercellular spaces. This tissue generally stores food. The cells have vacuoles and very small nucleus. Parenchyma again divided into 2 types. They are Chlorenchyma and Aerenchyma. Chlorenchyma. These cells contain chlorophyll and performs photosynthesis. So, this type of parenchyma called chlorenchyma. Aerenchyma. In aquatic plants, large air cavities are present to help them float. Such type of parenchyma tissue in plant is called as aerenchyma. Collenchyma. The flexibility in plants due to collenchyma. It allows bending of various parts of plants like tendrils and stem of climbers without breaking. It is also provides mechanical strength. We can find this tissue in leaf stalks below the epidermis. The cells of these tissue are living, elongated and irregularly thickened at the corners. These cells have very little intercellular spaces. Sclerenchyma. It is the tissue which makes the plant hard and stiff. We have seen the husk of a coconut. It is made up of sclerenchymatous tissue. These are dead tissues. They are long and narrow as the walls are thickened due to lignin. Often these walls are so thick that there is no internal space inside the cell. This tissue is present in stems, around vascular bundles, in the veins of leaves, and in the hard covering of seeds and nuts. It provides strength to the plant parts. Activity: section of a stem. Take a plant stem and cut into very thin slices. Now stain the slices with saffronin. Place one neatly cut section on a slide and put a drop of glycerin. Cover with cover slip and observe under the microscope. We observe various types of cells and their arrangement. Observations: all cells are not similar in structure. We can be seen variety of cells, that means epidermis, collenchyma, parenchyma, phloem, xylem, etc. Plants shows the many types of cells because they perform various functions. Activity: to observe God cells and epidermal cells. Take a freshly plucked leaf of Rio. Stretch and break it by applying pressure. While breaking it, some peel or skin projects out from the cut. Remove this peel and put it in a petri dish filled with water. Add a few drops of saffronin. Wait for 2 minutes and then transfer it onto a slide and place a cover slip over it. Observe under the microscope. Observations: the outermost layer of cells called epidermis, and it is made up of single layer of cells. In some desert plants, the epidermis may be thicker for protection against water loss in critical. The entire surface of plant has an outer covering, epidermis. It protects all the parts of the plants. Cells of the epidermal tissue form a continuous layer without intercellular spaces. We can observe small pores in the epidermis of the leaf. These pores are called stomata. Stomata are enclosed by two kidney-shaped cells called God cells. They are necessary for exchanging gases with the atmosphere. Transpiration, loss of water in the form of water vapor, also takes place through stomata. Epidermal cells of the roots have long hair like parts called root hairs. The main function of the root hair is absorption. In some plants, like desert plants, epidermis has a thick waxy coating of cutin on its outer surface. Because to prevent loss of water through transpiration or evaporation, the outer layer of a branch of a tree different from the other layer of a young stem. Because as plants grow older, the outer protective tissue undergoes certain changes. A strip of secondary meristem located in the cortex forms layers of cells which constitute the cork. Cells of cork are dead and compactly arranged without intercellular spaces. They have also a substance called suberin in the walls that makes them impervious to gases and water. Complex permanent tissue. Complex tissues are made of more than one type of cells. All these cells are coordinate to perform a common function. Xylem and phloem are the two different types of complex permanent tissues. Xylem. The main function of the xylem is transport of water from roots to all the parts of the plant. Xylem consists of tracheids, vessels, xylem parenchyma and phloem fibers. Tracheids and vessels have thick walls and many are dead cells when mature. Tracheids and vessels are tubular structures. They allows them to transport water and minerals. Xylem parenchyma stores food. Xylem fibers are mainly supportive in function. Phloem. The main function of the phloem is transport of food materials from leaves to all the parts of the plant. Phloem is made up of 5 types of cells. They are sieve cells, sieve tubes, companion cells, phloem fibers and phloem parenchyma. Phloem cells are living except phloem fibers.